Hello, I am Dr. Charu Dogra Rawat from Ramjas College, University of Delhi, and today I will be talking about the production of cloned and transgenic animals. This is a continuing series in biotechnology, and we had already talked about in a couple of lectures about the different methods for the production of cloned and transgenic animals. So, I will be continuing today talking about two more methods and some other technology. The learning objectives for today's session would be to understand the method of production of transgenic animals. As I said, in this we will be talking about another method, which is engineered embryonic stem cell method. We would understand the genetic modification by a Cre-Lox-P recombination system, and we would also be talking about understanding the method of production of transgenic animals, which is the somatic cell nuclear transfer or SCNT method. So, we have already talked about the different mechanisms beforehand, but this is again we will be talking about two more methods. The references used for the lecture, basically for educational purposes, is the T.A. Brown, the Gene Cloning and DNA Analysis 6th edition, as well as Glick and Pasternak and Patton, Molecular Biotechnology Principles and Applications of Recombinant DNA. Now, the objective of genetic engineering is to often create a useful product or a commercial process. When we say genetic engineering, we are making something. We are doing some modifications to the genomes or at the genetic level, at the DNA level and at other regulatory levels, and we are creating a useful product. So, one of the useful or application of genetic engineering is the production of cloned or transgenic animals. Now, what is a transgenic animal? A transgenic animal is one that has an integrated a gene or a DNA sequence, which is called as a transgene, that has been transferred by human intervention into the genome of a cell. Now, we have certain cells that we can modify it and express certain foreign genes in it. So, we can use these foreign genes, which are called as transgene, to express themselves and therefore create a modified version of the animal, which is called as the transgenic animal. A transgenic animal is defined as the one that has stably incorporated the transgene into its germline and is able to pass the transgene onto its offspring. So, this is one important aspect for the creation of transgenic animal, that it is not a transient expression, but that the foreign DNA or the transgene that we have introduced into the animal is stably incorporated and passed on to the offspring as well, that is, it is maintained in the germline. These are the different methods we have been talking about. So, we have already talked about the viral mediated gene transfer in our previous lecture. We were talking about the DNA microinjection method, in which we have talked about the pronuclear microinjection, in which the transgene is directly injected into the pronucleus of a zygote and then the embryo develops, the transgenic animal develops. Today we will be talking about the embryonic stem cell method, and we would also be talking about the stem cell nuclear transfer method or the SCNT method. These are the major mechanisms by which the transgenic animals can be produced. Now, let us look at what are embryonic stem cells. Embryonic stem cells are found in the inner cell mass of the human blastocyst, an early stage of the developing embryo lasting from the 4th to 7th day after fertilization. So, when the sperm and egg, they fuse together, they form a zygote, and the process is termed as fertilization. After fertilization, the human zygote undergoes cleavage, which is mitotic cell divisions, and then up 4th to 6th, 7th day, it forms a human blastocyst. Now, this human blastocyst has two different kinds of cells. One is a trophoectoderm and another is the inner cell mass. So, this inner cell mass are basically the embryonic stem cells. As they said, in normal embryonic development, they disappear after the 7th day and begin to form the 3 embryonic tissue layers. So, this inner mass basically give rise to the true embryo, and they form the 3 embryonic tissue layers or the 3 germ layers, giving rise to the ectoderm, mesoderm and endoderm, and then these 3 layers give rise to different organs and shape up the body. So, this human blastocyst, between the 4th to 7th day after fertilization, these inner cell mass can be isolated and used as the embryonic stem cells. Now, ESCs extracted from the inner cell mass during the blastocyst stage, however, can be cultured in the laboratory and under the right conditions will proliferate indefinitely. So, these are certain characteristics of ESCs which make them very useful for the production of cloned and transgenic animals. These ESCs, when extracted from the inner cell mass, they can be cultured in the laboratory. So, just in like plates, you can give the media, you can give the right conditions, and these ESCs will then divide and they will proliferate indefinitely. So, self-renewal property is there that they can produce more of themselves, and that too indefinitely, provided the conditions are met with. Now, ESCs growing in this undifferentiated state retain the potential to differentiate into cells of all the three embryonic tissue layers, a characteristic which is basically termed as pluripotent. So, potent, that is, it is able to differentiate, and pluri means it can actually differentiate into any of the cells of the body, being that of the ectoderm, endoderm or mesoderm. So, these ESCs, that is why they are called as the pluripotent cells, that they retain the potential to differentiate into cells of all the three embryonic tissue layers. Now, these two properties of self-renewal, that is the proliferation indefinitely, and the pluripotency, the two characteristics make them very useful for the production and cloning of transgenic animals. Now, let's look at how are the human embryonic stem cells derived. So, you can see in this slide that there are different mechanisms by which the transgene can be introduced. There is a simple method in which the designed fertilization, in vitro fertilization can be conducted. So, it is basically that a particular sperm is fused with the egg, or there are other mechanisms by which the transgene can be introduced into the egg. And then they will divide, as I said, in a normal developmental biology, they will divide, they will become 2-cell stage, 8-cell stage, then they will become a morula, and then a space is created, and as you can see, this is the blastocyst, where you have an outer ectodermal layer and you have an inner cell mass. Now, when I say that these embryonic stem cells, they are in this inner cell mass and they can be isolated. So, you can see that there are different methods by which they can be extracted. There are methods of immunosurgery, or there are methods of mechanical methods that can be used by taking out these inner cell mass, or even the entire blastocyst can be put on these feeder cells. These are irradiated feeder cells on the plate, mouse irradiated feeder cells, and then this inner cell mass can be proliferated on these feeder cells and grown and isolated, and then they can be put in these right conditions to differentiate them into any cell type. And then further we can grow them into form tissues or organoids or anything that will come on to in the later later lectures when we talk about the applications of this transgenesis that is done. So, the lower part of this slide is basically telling you what I have been talking about for the generation of the embryonic stem cells. Now, the problem is that, it's not a problem, but that is basically an ethical and political issue that comes in here, is that when we extract the inner cell mass, then this blastocyst is discarded. So, that is basically going to develop into an embryo, but it is now discarded because we take out the inner cell mass and we grow it. So, this raises a lot of ethical concerns. So, sometimes what happens is that the cells are isolated from the stages before this blastocyst. So, some of the cells, they can be extracted from the 2-cell, 8-cell stages, and they can also be grown onto these plates containing feeder cells, or from the morula as well. These cells can be extracted and grown. So, this, and then they continue to form the blastocyst. So, some of the cells are extracted, grown into plates, and then it continues to grow just like a normal embryo. So, again it depends, but the classical transgenesis involved the derivation of human embryonic stem cells from the blastocyst, the inner cell mass, and that was basically as it is written here, can be extracted by immunosurgery or by mechanical thing. One thing is the ethical concerns, another thing is that this immunosurgery involves the use of certain animal products, certain animal hormones, and in fact here also we are using irradiated feeder cells, the mouse feeder cells. So when we provide certain animal products or hormones, etc., it has the influence of these animal products, which is basically said to be the xeno effects, and therefore these cells cannot be transplanted back into the humans or to generate the transgenic clones that we want. So that is also one of the problems, and that is why sometimes these cells are taken from the previous stages, which can counter the use of the feeder cells as well, and therefore it can be a xeno-free. So many xeno-free mechanisms have also been developed to derive the human embryonic stem cells, but this is the basic mechanisms that we need to talk about. Now, as I said, this stem cell research, the generation of the pluripotent stem cells, these stem cells can be employed to form, the tissue and cell therapy, and everything can be done by generation of different organoids or different tissues such as bone marrow, nerve cells, heart muscles, etc. They can be induced to develop into any of the tissues and therefore can be used in the therapy. They can also be used for developmental research, that how does the development occurs, what are the key transcription factors or growth factors, etc., that are stimulated, or how the different types of growth can occur. And then they can also have different kind of developmental stages, and different research for the tissue generation, etc., can also be done. So this stem cell research, this field of biotechnology is a very proliferating field, and in fact used for various kinds of gene therapies and different kinds of disease treatments, as well as preventions, and simultaneously helping in progressing the developmental research which can be done on these stem cells. So, these were the embryonic stem cells, and how to derive these embryonic stem cells. I particularly took the example of human embryonic stem cells, but similarly the embryonic stem cells can be isolated from any of the animals. But the main important part in biotechnology is that how do we engineer these ESCs? Because if we want to manipulate at the genomic level and we want to express certain features which are of our desired interest, then how these ESCs can be engineered. So this engineered ESCs is a method used for the production of transgenic animals. Now, engineered ESCs is a technique where scientists manipulate the genetic makeup of ESCs by introducing specific genetic modifications, typically through techniques like homologous recombination, to create ESCs with desired characteristics for research or potential therapeutic applications. Now, important point here is that the scientists manipulate the genetic makeup. So, different kind of genetic modifications are done, insertions, deletions, inversions, or knockouts, knock-ins. All these kind of modifications are done at the genomic level, and the techniques that we will discuss also is used, such as homologous recombinations, so that the ESCs with the desired characteristics are created. And these desired characteristics can be for the generation of a better adapted animal, a transgenic animal, or plants, or for certain therapeutic applications such as to do the therapy for the curation of a certain disease or certain abnormal conditions or whatever. So that is basically the engineered ESCs are generated for. Now, the method involved for the creation of transgenic mice, The first is the gene targeting of the embryonic stem cells. As I said, we have discussed about how the embryonic stem cells can be generated. So, from the 4th to 7th day inner cell mass, we put them on the plates, and we proliferate these ES cells. Now, these ES cells can be targeted by, so as it is shown, these are the mouse blastocyst, and the cells are taken. Now, these can be targeted by the vectors that contain our transgene or the gene of interest. So, this transgene is basically the foreign DNA that we want to introduce for certain characteristics to be there, and they can be transfected. So, there are different methods we have already talked about, that how these vectors can be introduced into the cells, the bacterial cells or the eukaryotic cells. So, they can be like, for example, chemically transfected, or there can be physical methods such as electroporation. So, these vectors containing the desired gene of interest can be transfected into the embryonic stem cells, and certain selections can be done so that the modified or engineered ES cells are obtained. So, this is the first step for the creation of the transgenic mice, that the engineered or modified embryonic stem cells are prepared. The second step involves the integration of these. So, these, in fact, the engineered stem cells, they can be introduced back into the blastocyst, and then this can develop into or this can be put into a surrogate mother. So, this blastocyst can then be implanted into the surrogate mother, which will give rise to the chimeric mice. You can see the different mice which will have the genome, a double genome where you have this transgenic expressed somewhere. Now, when these chimeras are crossed with the wild type females, they are going to give rise to a pure line of transgenics and the pure line of the normal mice which are there. So, that is how basically the pure line of transgenics can be generated. Now, this has been widely used to create the transgenic mice, and then the presence of the transgene can be checked by taking a tip of the tail and then doing the genotyping. So, you can take the tip of the tail and dissolve and isolate the DNA and do the PCR amplification of the transgene that you have introduced. So, this pure line of transgenics will be now incorporating and establishing and expressing the transgene that you have introduced. So, this is basically the mechanism of creating a transgenic mice using the engineered ESC method. So, just a quick review: the blastocyst, take out the inner embryonic stem cells, maintain that, proliferate them, then transfect them with a vector containing the foreign DNA or the transgene, select the modified ESCs, then implant them back into a blastocyst, introduce them into a surrogate mother, and then get the progeny. These progeny will be the chimeras. Chimeras means expressing two different types of characteristics for a particular trait. So, these are the chimeras at the genetic level, and then you crossbreed this with the normal wild type females, and you will get a pure line of the transgenic as well as these other normal mice as well. So, this transgenics, then you can do the genotyping by taking the tail tip and isolating the DNA and PCR amplification to find out whether the transgene is expressed or not. So, this is an engineered ESC methods. Now, an important aspect is that how does this transgene that you have introduced into the embryonic stem cells, that you transfect into the embryonic stem cells, gets integrated into the genome. That is an important aspect, and the major mechanism that is found is the homologous recombination. So, the vector that you have introduced carrying your transgene will, this is for example the vector, it will homologously recombine with ES cells, and in this case, for example, the thymidine kinase is introduced into it, as well as there is a resistant marker, which is the neoresistant marker. So, neoresistance will be introduced. So, the host is neosensitive, and as I said in this particular case, that when you do this selection for the stable transgenic embryonic cells, then in this case, for example, you have got a neosin resistant marker. The embryonic stem cells which will be neomycin resistant will be the one which are the modified ES cell. So, this is a straightforward thing where we can do the where the homologous recombination is occurring. The targeted transgene, the transgene is introduced at the targeted site, for example, the introduction of this thymidine kinase along with a marker, resistant marker, so that the modified ES cells can be selected, and then this modified ES cells can be introduced into the blastocyst and then to the surrogate mother and give rise to the transgenic progeny. So, homologous recombination is an important mechanism for the integration and stable expression of the transgene in the genome of the host. There is another thing which we generally do, which is called as the positive and negative selection. So, this positive and negative selection is also an important aspect in which the target vector contains the gene of interest or whatever, this thymidine kinase for example, and the neomycin resistant, but they are not together, they are a little far apart, because we also have to understand that the integration has occurred at the target site only, because this integration, we do not know whether this integration will be due to the homologous recombination or this integration is a random integration. So, we do not want the random integration, we are not interested in the embryonic stem cells where the random integration would occur, we would only be interested in the embryonic stem cells where the targeted introduction has occurred. So, in this case, we can do a positive negative selection in which we take these one gene and a resistant marker. Now, if there is a gene targeting, that is the target vector introduces at a targeted site, then the homologous recombination will occur. Now, because for the homologous recombination, this thymidine kinase is outside the homologous recombination region. So, when the recombination occur, only the neomycin resistant marker will be introduced into the target gene, so that we will select those colonies which will be neomycin resistant positive. Now, if there is a random integration and there is no homologous integration, then this entire stretch will get introduced into the target, into the genome, into the region, and when it is introduced, it will be neomycin resistant positive as well as thymidine kinase positive. So, with the neomycin resistant positives, we can differentiate between the ones which are gene targeted, which will be thymidine kinase negative, and the just random integration, which will be thymidine kinase positive. So, this is a positive negative selection which can be used to identify whether the gene targeting, the introduction has occurred because of the gene targeting or has occurred because of the random integration. So, as I said, we require the modified embryonic stem cells, but only those where the homologous recombination has occurred. Two reasons: one, because that will be stable; second, because it is at the targeted site that we want it to be introduced to. If there is a random integration, then that random integration can be not very stable, one thing, and second, because it has been introduced randomly. So, we don't really want that. We want it to be targeted at a particular gene. So, this positive negative selection of the homologous recombinants is very useful to get the modified ES or the engineered embryonic stem cells, and then this engineered embryonic stem cells can be introduced into the host and into the mice. For example, the blastocyst, we plant them into the blastocyst, we introduce them into the mice and can generate the transgenic mice. So, the production and cloning of transgenic mice, Nobel Prize in Physiology or Medicine in 2007 were given to these scientists, the great scientists Cappucci, Evans and Smithies, and this was given basically awarded jointly to Cappucci, Sir Martin J. Evans and Oliver Smithies for their discoveries of the principles of introducing specific gene modification in mice by the use of embryonic stem cells. So, this is a remarkable discovery that has been there. In our next session, we will talk about another technology for the gene modification that can be done to generate the engineered embryonic stem cells. Thank you.